A cytotoxic T-cell, also known as TC, cytotoxic T lymphocyte, CTL, T-killer cell, cytolytic T-cell, CD8 plus T-cell or killer T-cell, is a T lymphocyte, a type of white blood cell, that kills cancer cells, cells that are infected, particularly with viruses, or cells that are damaged in other ways. Most cytotoxic T-cells express T-cell receptors, TCRs, that can recognize a specific antigen. An antigen is a molecule capable of stimulating an immune response, and is often produced by cancer cells or viruses. Antigens inside a cell are bound to class I MHC molecules, and brought to the surface of the cell by the class I MHC molecule, where they can be recognized by the T-cell. If the TCR is specific for that antigen, it binds to the complex of the class I MHC molecule and the antigen, and the T-cell destroys the cell. In order for the TCR to bind to the class I MHC molecule, the former must be accompanied by a glycoprotein called CD8, which binds to the constant portion of the class I MHC molecule. Therefore, these T cells are called CD8 plus T cells. The affinity between CD8 and the MHC molecule keeps the TC cell and the target cell bound closely together during antigen-specific activation. CD8 plus T cells are recognized as TC cells once they become activated and are generally classified as having a predefined cytotoxic role within the immune system. However, CD8 plus T cells also have the ability to make some cytokines. Development. The immune system must recognize millions of potential antigens. There are fewer than 30,000 genes in the human body, so it is impossible to have one gene for every antigen. Instead, the DNA in millions of white blood cells in the bone marrow is shuffled to create cells with unique receptors, each of which can bind to a different antigen. Some receptors bind to tissues in the human body itself, so to prevent the body from attacking itself, those self-reactive white blood cells are destroyed during further development in the thymus, in which iodine is necessary for its development and activity. TCRs have two parts, usually an alpha and a beta chain. Some TCRs have a gamma and a delta chain. Hematopoietic stem cells in the bone marrow migrate into the thymus, where they undergo VDJ recombination of their beta chain TCR DNA to form a developmental form of the TCR protein, known as pre-TCR. If that rearrangement is successful, the cells then rearrange their alpha chain TCR DNA to create a functional alpha-beta TCR complex. This highly variable genetic rearrangement product in the TCR genes helps create millions of different T cells with different TCRs, helping the body's immune system respond to virtually any protein of an invader. The vast majority of T cells express alpha-beta TCRs, alpha-beta T cells, but some T cells in epithelial tissues, like the gut, express gamma-delta TCRs, gamma-delta T cells, which recognize non-protein antigens. T cells with functionally stable TCRs express both the CD4 and CD8 CO receptors and are therefore termed double-positive, DP, T cells, CD4 plus CD8 plus. The double-positive T cells are exposed to a wide variety of self-antigens in the thymus and undergo two selection criteria. Positive selection, in which those double-positive T cells that bind to foreign antigen in the presence of self-MHC. They will differentiate into either CD4 plus or CD8 plus depending on which MHC is associated with the antigen presented, MHC1 for CD8, MHC2 for CD4. In this case, the cells would have been presented antigen in the context of MHC1. Positive selection means selecting those TCRs capable of recognizing self-MHC molecules. Negative selection, in which those double-positive T cells that bind too strongly to MHC-presented self-antigens undergo apoptosis because they could otherwise become autoreactive, leading to autoimmunity. Only those T cells that bind to the MHC self-antigen complexes weakly are positively selected. Those cells that survive positive and negative selection differentiate into single positive T cells, either CD4+ or CD8+, depending on whether their TCR recognizes an MHC class I presented antigen, CD8, or an MHC class II presented antigen, CD4. It is the CD8+ T cells that will mature and go on to become cytotoxic T cells following their activation with a class I restricted antigen. Activation. With an exception of some cell types, such as non-nucleated cells, including erythrocytes, class I MHC is expressed by all host cells. When these cells are infected with a virus, or another intracellular pathogen, the cells degrade foreign proteins via antigen processing. These result in peptide fragments, some of which are presented by MHC class I to the T cell antigen receptor, TCR, on CD8+ T cells. The activation of cytotoxic T cells is dependent on several simultaneous interactions between molecules expressed on the surface of the T cell and molecules on the surface of the antigen-presenting cell, APC. For instance, consider the two-signal model for TC cell activation. A simple activation of naive CD8+ T cells requires the interaction with professional antigen-presenting cells, mainly with matured dendritic cells. To generate long-lasting memory T cells and to allow repetitive stimulation of cytotoxic T cells, dendritic cells have to interact with both activated CD4+ helper T cells and CD8+ T cells. During this process, the CD4+ helper T cells license the dendritic cells to give a potent activating signal to the naive CD8+ T cells. While in most cases activation is dependent on TCR recognition of antigen, alternative pathways for activation have been described. For example, cytotoxic T cells have been shown to become activated when targeted by other CD8 T cells, leading to tolerization of the latter. Once activated, the TC cell undergoes clonal expansion with the help of the cytokine interleukin-2, IL-2, which is a growth and differentiation factor for T cells. This increases the number of cells specific for the target antigen that can then travel throughout the body in search of antigen-positive somatic cells. Effector functions. When exposed to infected/dysfunctional somatic cells, TC cells release the cytotoxins perforin, granzymes, and granulysin. Through the action of perforin, granzymes enter the cytoplasm of the target cell and their serine protease function triggers the Caspase cascade, which is a series of cysteine proteases that eventually lead to apoptosis, programmed cell death. A second way to induce apoptosis is via cell surface interaction between the TC and the infected cell. When a TC is activated, it starts to express the surface protein Fos ligand, Fos-L, APO1L, CD95L, which can bind to Fos, APO1, CD95, molecules expressed on the target cell. However, this Fos-Fos ligand interaction is thought to be more important to the disposal of unwanted T lymphocytes during their development or to the lytic activity of certain Th cells than it is to the cytolytic activity of TC effector cells. Engagement of Fos with Fos-L allows for recruitment of the death-induced signaling complex, DISC. The Fos-associated death domain, FADD, translocates with the DISC, allowing recruitment of procaspases 8 and 10. These caspases then activate the effector caspases 3, 6, and 7, leading to cleavage of death substrates such as Layman A, Layman B1, Layman B2, PARP, poly-ADP ribose polymerase, and DNAPK, DNA-activated protein kinase. The final result is apoptosis of the cell that expressed Fos. Role in disease pathogenesis. During hepatitis B virus, HBV, infection, cytotoxic T cells play an important pathogenetic role. They contribute to nearly all of the liver injury associated with HBV infection and, by killing infected cells and by producing antiviral cytokines capable of purging HBV from viable hepatocytes, cytotoxic T-cells also eliminate the virus. Platelets have been shown to facilitate the accumulation of virus-specific cytotoxic T-cells into the infected liver. Cytotoxic T-cells have been implicated in the progression of arthritis, depletion of knee joint cartilage macromolecules such as glycosaminoglycans by cytotoxic T-cells and macrophages has been observed in a rat model of the disease.
Please subscribe and thanks for watching.